So welcome everyone to this week's Autonomy Talk. This week is a great pleasure to have with us Professor Vickie Webster-Wood, who is an associate professor in the Department of Mechanical Engineering at Carnegie Mellon University, where she also has courtesy appointments with the Department of Biomedical Engineering and the McGowan Institute of Regenerative Medicines and the Robotics Institute. So something about Vickie. Vickie obtained her PhD and then completed a postdoc at the Case Western Reserve University. And at CMU, she directs the Biohybrid and organic robotics group, where the general research brings together bioinspired robotics, tissue engineering, computational neuroscience, and in general, in her lab, people want to study and model muscular control and translate findings to the creation of renewable robotic devices. So this is a super exciting area that we don't hear much about in this series, and I'm really excited to hear more about it. And the talk today is titled Biomimetic, Biohybrid, and Biodegradable Robots for a Sustainable Future. And without further ado, Vickie, go ahead. The stage is yours.
Thank you so much for the great introduction and for inviting me to speak with you all today. Hopefully this isn't too far afield from the typical kind of theme of this talk series, but I hope to kind of stimulate some really excellent discussion around this idea of biomimetic and biocompatible robotics. So it's my great pleasure to be with you for this seminar and to be able to share research that's going on in my lab. As was said, I run the Biohybrid and Organic Robotics Group at Carnegie Mellon University, and we have been conducting research in biomimetic, biohybrid, and biodegradable robots towards a long-term goal of creating completely autonomous, sustainable robots in the future. And before I dive in, I of course want to take a moment to acknowledge the Borg team. The work I'm sharing today would not have been possible without this amazing crew of student and postdoctoral researchers, as well as our research technicians and our wonderful alumni through the years. So to get us started, in my lab, we often start by looking at animals. And animals are capable of tremendous feats of agility, strength, ingenuity, creativity, and learning. They adapt and respond to dynamic changes in their environment. They learn from experience, and they solve complex real-world problems. As an engineer, these are the capabilities that we want to achieve in future robotic systems. So towards capturing these capabilities, in my lab, as I said, we focus on biomimetic, biohybrid, and biodegradable robots. We want to be able to learn fundamental principles of actuation and control by studying and modeling biological organisms, and ultimately even harness the material capabilities of living systems for future robotic solutions. We do this by identifying and characterizing renewable biocompatible materials and appropriate fabrication techniques. And then we use these materials to build adaptable living actuators and energy efficient living controllers. And the ultimate goal in my lab is to create soft, bio-inspired robots that can themselves safely interact with us and our environment. And in the future, we hope that these may one day even be made entirely of materials that are biodegradable and renewable, and that the use of these robots will then allow us to better understand the biological organisms that inspired us initially. So for today's talk, I'm going to share characteristic examples of our work in these areas. This will include our work in biomimetic robots as tools for basic neuromechanics research. And these have led to bio-inspired robots for applications in spaces like agriculture. From there, I'll discuss how we are building foundational tools in biohybrid robotics, including kind of materials modeling and manufacturing capabilities. And I'll wrap up with some of our more recent work in creating biologically sourced marine-derived biodegradable actuators. So let's dive in. One of the key questions that drives the research in my group is how can animal principles allow robots to generate multifunctional adaptable behavior? We come at this from a very bio-inspired standpoint. So to address this question in bio-inspired robotics, we want to try to use neuromechanical models and robots in order to actually capture animal behavior. for this, we need a tractable animal model that does generate that type of adaptable behavior. And so we turned to the sea slug, Aplysia californica, which is the funky critter that you're seeing here. Aplysia is a robust marine sea slug, and it has a relatively small nervous system. So in particular, we typically study the feeding behavior as a model of soft grasping, and the ganglia, the clusters of neurons that control feeding behavior, between the two of them probably only have about 4,000 neurons. So it's very feasible that we could actually study and model many, if not all, of the neurons in this structure. And as such, it's been a model system in neuroscience for decades. It has a completely soft-bodied musculature. There's no skeletal system, so it's also an interesting mechanical problem, and it demonstrates multifunctional behavior during feeding. This includes biting, which is an attempt to grab seaweed. That's what you're seeing in this video here. There's also swallowing, in which that grasper closes on seaweed and pulls it in. And there's rejection, in which the animal is able to spit out inedible food. And so with this all in mind, we look at the components of the system. These behaviors are all generated by a single soft feeding apparatus called the buccal mass, located in the head of the animal. And the muscles within this structure are controlled by the buccal ganglion, which we can kind of think of as a motor controller, and this receives higher level commands from the cerebral ganglion. We can break the problem of multifunctional behavior kind of into two sections. First, we need to understand what neural units are needed to model multifunctional neural behavior. And second, we need to understand how the musculature itself is contributing to this behavior through morphological computation. For the control model, as we were developing this work, our goal was really to create a faster than real-time model that could predict multifunctional neural control patterns and generate that behavior in a biomechanical model. So to achieve this goal, we of course went and looked at the actual neural circuitry of Aplysia. That was our tractable animal model, and we needed a computationally efficient modeling framework. So we created a hybrid Boolean continuous model of this feeding behavior, and I'll get kind of into the details of what that looks like next. To start, we assess the key neurons and muscles involved in feeding behavior on an individual basis. And we do this in close collaboration with neuroscientists who are actually doing these recordings in the animals. For example, in swallowing, there are kind of 4 phases of behavior that are observed. This starts with pinching, in which case the front of this feeding apparatus, the jaws, closes on the seaweed in order to hold the food. Then that grasper apparatus that we saw in the videos is protracted forward, where it closes, and then it's able to be retracted back and push the food into the esophagus. And this cycle repeats, thereby generating force on the seaweed. And so our neuroscience collaborators can go implant electrodes in the animal and actually collect this neural recording data, either through EMG recordings with hook electrodes on the muscle or with directly implanted probes in neurons or hook electrodes on the nerves that those neurons innervate. And we can approximate this neural activity in each of these boxes as a Boolean burst, right? So the neuron can be considered as either off, represented by 0. This is when the neuron is quiescent, or on, represented by 1. That's when the neuron's firing. So we created this network based on this idea of approximating firing as these Boolean bursts. To build the network, we start with our team's prior work and the existing literature, and we build the network in layers. First, we create this motor neuron layer based on the key neurons we identified in our recordings. Next, we build an interneuron layer. These kind of coordinate the activity of the motor neurons. And then we build a cerebral interneuron layer, where these are kind of like those higher level commands that control behavioral switching. And each of these neurons is a known unit in the slug's nervous system that we can consistently identify experimentally. External sensory cues are applied to the cerebral interneurons. This gives kind of context of what feeding state we want to be in. and the motor neurons project onto a simulation of the biomechanics of the feeding apparatus, which then provides sensory feedback, and that's input into the interneuron layers. So with this model, we're then able to run kind of predictive simulations, and we find that the model qualitatively reproduces the three key multifunctional behaviors. So for example, with biting, this occurs in the animal when there's no mechanical stimuli in the grasper, right? So it hasn't grabbed seaweed yet, but there's sensory cues that signal seaweed is present in the environment. And when we simulate this combined neural biomechanical model, we see that that grasper moves through a protraction-retraction cycle with retraction when the grasper is closed, just as we see in the animal. It also produces swallowing just by changing the sensory stimuli. So here, if there's mechanical stimuli in the grasper, that means that, yes, there's seaweed in the slug's mouth, and so it's going to try to swallow. And swallowing is characterized by this strong retraction as force is applied to the object. The model also reproduces rejection. So if there's no chemical stimuli, then it probably means the thing you're holding onto is not seaweed. And so the animal then is able to push the seaweed out with this strong rejection force. Interestingly, just by mimicking the structure of the nervous system and approximating the biomechanics, we also get emergent behaviors that are not necessarily inherently programmed or hard-coded into our model. For example, the model can produce behavioral switching. So if there's a sensory state change, in this case from a biting-like state to a swallowing-like state, we see that change in the behavior. And it can produce multifunctional behavior even within a single one of these behaviors, which we tested by simulating seaweeds of varying strengths. So if we simulate a weak seaweed, we see that the grasper moves through that protraction-retraction cycle. The force on the seaweed during retraction starts to rise until you reach a force threshold that we've specified, and then the seaweed breaks. And this behavior with the weak seaweed looks much more like a biting pattern, just like we see in the animal. And in contrast, if we simulate a strong seaweed, we again get that retraction-protraction cycle. But here now the force on the seaweed remains high until the grasper releases the seaweed. And there was basically no threshold values or cutoffs or anything like that programmed into the model. It's just kind of an inherent property of the intrinsic dynamics of the nervous system and the sensory feedback pathways that these types of multifunctional behavior emerge. Okay, so that's very animal centric, right? And in our initial neuromechanical models, we implemented a very simple approximation of the biomechanics. Really, we just modeled the grasper in the head as two point bodies connected by springs to represent the elasticity of the musculature. Which it was a very pleasant surprise that even those types of simple approximations with these neural models are able to generate interesting behaviors. But our goal is really to understand how to leverage these types of controllers in the control of robots. And so we also want to better represent the mechanics of the system. But this is just a subset of all of the muscles that are present. You might well imagine that this is very complex to simulate and even understand. And getting all of the soft body context physics is challenging. And so this is where we start to turn to robotics. And we can build in roboto models of the feeding apparatus in order to allow us to test biological hypotheses of control and identify which features are really kind of helping generate that multifunctional behavior so that we can translate them to other robotic applications. In this robotic model, we include physical representations of imported feeding muscles and structures. So for example, the primary retractor muscle, I3, and these external I1 muscles are mimicked using McKibben-style pneumatic artificial muscles. And these are fabricated using an internal bladder housed inside an overexpanded mesh sleeve. Of course, the mesh restricts the motion of the actuator during expansion, thereby causing contraction along the axis of the actuator. The I1 muscle uses kind of your standard linear McKibben design, and the I3 muscle was approximated as a series of toroidal rings. In order to achieve larger contraction ratios that we needed in order to mimic the sea slug's muscles, we actually fabricated these in-house using a latex balloon as the internal bladder, because it allows us to get really good contraction ratios with relatively low pressure. And we just adhere the latex balloon to these plastic fittings with Kevlar thread and cyanoacrylate in order to create a really strong seal. For the primary protractor muscle, I2, this is a very interesting muscle. It's kind of a broad Y shape, and it's exceptionally thin. So to mimic this geometry and the fiber orientations that we needed in the muscle, we created arrays of flat artificial pneumatic actuators by modifying a technique from Weirko et al. So they used dissolvable PVA films and reinforcing Kevlar threads to create flat pneumatic actuator units, like the ones that we fabricated here. And so we adapted this to our application by creating a stacked mold approach where we sequentially assemble these molds in order to cast different layers of elastomer and embed these reinforcing polyester films, which were just cut with a silhouette portrait 2 in order to get the fiber orientations that we wanted. So we lay this all up with Smooth On Dragon Skin 10, and then once that's cured, we can then wash out the PVA film using deionized water. For the grasping apparatus, we built this kind of internal mechanism that is also actuated by linear McKibben actuators. And we replicate the kind of soft jaw structure of the sea slugs using this reinforced rubber sheeting, which goes over a force sensor so that we can detect that sensory input at the jaws. And then we bring all these pieces together. And if I can get the video to play, we're then able to connect the robot to that model of the sea slug's neural system to generate behavior. So here you see the grasper rotating forward. It's going to grab onto this tube, which is representing the seaweed in this case. And then the I3 will actuate and push that grasping apparatus backwards such that the seaweed is deposited kind of out of the jaws. And of course, it moves very slowly. Sea slugs move very slowly. So we do dynamically scale all of our models to make sure that we're moving in the same dynamical regime. So we first compared the robot's performance in both biting and swallowing to the performance of our original simulation and found that the robot does produce qualitatively similar behaviors. But more importantly, we're now able to compare the robotic performance to the actual animal data. And we do this both for biting, looking at the translation of that grasper, and for rotation. And we find that both for biting and for swallowing, there are a lot of similarities in the behavior of the robot and in the behavior of the animal. And yet, there are differences in timing, there's differences in amplitude that really point to additional research hypotheses about what features of the musculature or the nervous system are critical for this behavior. And so then this creates this kind of feedback loop with the neuroscientists to keep iteratively identifying new features both in the animal and applying those to the robots. So this allows us to use these kind of biomimetic robots in basic neuroscience research, but we also start seeing areas of inspiration for more general soft grasping robots, right? In the sea slug, of course, we have this buccal mass, we have the buccal ganglion, and we have the cerebral ganglion, and we can think about these from a very biological standpoint. But we can also abstract these to see how they might apply more broadly in robotics. For example, you can consider the muscles as viscoelastic adaptable actuators. You can consider the nervous system as an adaptable controller capable of learning. There's the presence of time history dependent actuator control through neuromodulation. And we've got these jaws with tunable stiffness. And so based on these principles, we are now developing engineering systems that capture each of these features. So we abstract this onto a soft grasper. And for this initial prototype, we took inspiration from the radial contraction of the I3 and the tunable stiffness of the jaws. And we can, again, use similar techniques to our biomimetic robot in order to fabricate this soft grasper. And you can see this all assembled here. And so we can tune both the displacement of the kind of circumferential actuator and the stiffness of the jaws by varying the pressure in the system. To control this, we're again taking inspiration from computational neuroscience, and we use an approach called synthetic nervous systems. So these basically capture bioinspired network dynamics using a rate-based model of the neuron. Unlike traditional ANNs, each neuron captures sorry, my headset turned off. Biologically plausible dynamics by modeling an axonal and a soma membrane with conductance-based ion channels and synapses. The original representation of synthetic nervous systems, if you go into the literature, is a continuous time representation, but we're now working on discretizing this. So using these neural units, we can build a grasping controller taking inspiration from the hierarchical structure of the sea slug nervous system without having to mimic every single neuron. And excitingly, the controller tuned in simulation in this way actually quite easily transferred to the physical hardware with minimal retuning. And so you can see again, if I play these, the performance of the simulation and the grasper when under control of this system. This grasper is capable of pick and place operation with a wide variety of objects, including really fragile objects. So now we've tested obviously with eggs, as all soft grasping robot people do. We've also tested with tomatoes and avocados, and are working on doing tests with really soft clay currently to really understand the damage and deformation. And what we also see is that even with these kind of bio-inspired features, there's really a kind of a trade-off, right, between grasping stability and damage on the object. And if you try to take kind of a naive approach to this and set force thresholds or position thresholds, you're likely to bruise your very fragile agricultural objects. So inspired again by the sea slugs, we take inspiration from neuromodulation, which you can think of as a time history dependent adaptation. And so in the animal, we see this as, stimulation of certain muscles just at a very low, almost sub-threshold level, ultimately leads to stronger contraction of subsequent stimulations. We can mimic this with our synthetic nervous system neurons and apply this to our hierarchical neural controller. In particular, we add neuromodulation in the jaw contraction, as well as some feedback pathways in the rest of the nervous system. Taking this adaptation first to our simulation for both position and velocity type control, we see that this really improves grasping performance. So here, without adaptation, with using a velocity controller to avoid high contact forces, we get about a 50% success rate in grasping. But when we allow adaptation, the success rate improves dramatically. And you can see that here, where as compared to position control, velocity control without adaptation, as the objects get heavier and heavier, leads to higher failure rates, whereas velocity control with the adaptation maintains a very high success rate while having a much lower maximal force on the object. We also tested this on the physical system. And here you can see some examples of grasping with and without adaptation. So without adaptation, the grasper tries to repeatedly pick up this heavy object and just continues to fail over and over again. And all 10 trials without adaptation failed. But with adaptation, the grasper is able to slowly adapt its force and be able to pick up heavy objects without a priori knowledge of the weight of the object. So we see this in our physical experiments. Here you can see the jaw pressure of the network without adaptation. And so for all of its trials, it really fails to get a good grip on the object. Whereas with adaptation, you can see that jaw pressure increases in subsequent trials, and many of these trials stop well below our trial number threshold because they have successfully lifted the object. Most recently, just as a little teaser, we've also been working on discretizing the synthetic nervous system network so that we can use more standard ML techniques like backprop to train the network and learn parameters. And in some initial prototypes that we've done, just trying to train specific functional subnetworks, we find that the SNS, when in discretized form, is able to learn appropriate parameters for nonlinear relationships with a very sparse network, much faster than the same size ANN. So that's super cool. Okay, so that gets us kind of through the biomimetic side of the lab. And I do want to spend a little bit of time on the biohybrid and biodegradable side of the lab. So as we all know, kind of an unfortunate reality facing us in the coming century is that natural disasters and supply chain interruptions are only likely to increase. These supply chain disruptions impact us as researchers as it becomes more challenging to obtain the parts we need to build robots. But we also need to consider the downstream effects of our robots. So after deployment, what happens to our robots? If we step back and think about this, we can start to think about the impact of our research on future generation of potentially harmful products. So after deployment, robots may break, they may leak, they may be used and discarded. A student works on a project for 4 years, they graduate, they move on. Maybe the luckiest of the robots may end up recycled for their parts and used again in another robot or in a museum. And ultimately, if we're doing our job right, our robots are supposed to become obsolete. Now, I of course acknowledge that consumer electronics, our phones, our laptops, our cameras, these have a far greater impact on the electronic supply chain and the growing piles of e-waste. But since our job as researchers is so often to think forward and consider the future of our field, I pose to all of you a challenge to think about the long-term environmental effect of our research. In contrast, animals are composed of sustainable biodegradable materials that are adaptable to their environment, that already have many of the features that we look for in robotic systems. And so one of the things we do in my lab is we look at how we can leverage the desirable properties of living materials directly in robotics. We consider this in the materials themselves, in the structures, in the controllers, and in the actuators. And although this idea of biohybrid robotics has received growing interest over the past several years, there are challenges that remain in adapting these approaches. In particular, these include current limitations in the types of robots that can use muscle-based actuators because of the very soft, compliant nature of muscle. This includes high barriers of entry for new researchers and the lack of kind of biological controllers that can really be predictably programmed. So we're making some small strides in these spaces. Starting off, I'll just highlight some of our work in manufacturability of ECM-based components. So one material that shows a lot of promise for biohybrid robotics is electrocompacted and aligned collagen. We fabricate this material by introducing a collagen solution, just isolated from tendons typically, between two electrodes. And by applying a current across these electrodes, the collagen molecules become charged and they compact along the isoelectric point. Using this technique, we can fabricate really robust collagen threads and sheets, and with chemical cross-linking, we can get their strengths even up to approaching those of sutures. What's really exciting about this material is that the alignment of the collagen can also be used to align muscle cells that are seated on the sheets. We've used this approach previously to create little tiny, these are like millimeter scale collagen robot skeletons. We seed these with muscle cells, and then under the passive stresses of the cells, these robots take on a 3D shape and can be stimulated. And they'll crawl very, very slowly. I promise this robot is moving. These early prototypes with these materials really indicated that there are some limitations when working with biohybrid actuators. And these include kind of limited deflections and low muscle forces when using monolayer cultures. So to overcome these, we started by looking at the skeleton material. And there's a lot of manufacturing knobs that we can tune when working with this electrocompacted collagen. Including the kind of time of compaction, the voltage, the concentration of the collagen. And what we were interested in is how does kind of the factorial modification of these manufacturing parameters affect the final geometry and mechanics of the threads. So I'm not going to go through all the study results, but if you're interested, this work was published a number of years ago. Some of the key takeaways include that ELAC fibers made with longer IF durations showed significantly higher tensile strength, failure strain, and a larger cross-sectional area. And ELAC fibers made with lower voltage exhibited significantly lower tensile strength, modulus, and cross-sectional area. And interestingly, we saw joint effects of these manufacturing parameters. Together, these allow us to really kind of have a wide range of mechanical properties that we can achieve with these materials, and thereby allow us to kind of control the resulting properties of any cells that we're going to grow on them. So the other kind of challenge facing some of those early biohybrid robots was that muscle, when you grow it just as a single layer, forms really immature muscle tissue. It is not nearly as strong as mature adult muscle tissue. Recently, we've seen a lot of work moving into 3D culture of these muscle tissues, and this shows a lot of promise. So we've moved from these kind of flat cantilever style robots into 3D printed robots with these 3D muscle tissues. These are typically cast around the 3D printed structure, or some of them now are being bioprinted in order to create the geometry of the actuator. And these are all relatively small scale. The robots themselves are relatively soft. And there's a lot of challenges in interfacing these types of muscles with a robotic structure, just because of the mismatch in the mechanics, right? So if you try to connect a really soft muscle to a rigid structure, tears are common, right? We're lacking that kind of mechanical gradient that we have in our actual skeletal musculosystem. So this brings us into some work that we're doing towards manufacturing reinforced structures for these biohybrid robots. In particular, we're working in the bioprinting space. And we have been collaborating with Dr. Adam Feinberg, who developed the freeform reversible embedding of suspended hydrogels technique in order to integrate fiber reinforcement into bioprinted muscle. In this fresh printing technique, you extrude your bioink into a shear thinning support bath, and this allows you to print very small, very intricate structures, even with overhangs, out of hydrogels, which would otherwise be too soft to hold their own weight. And so you can see here kind of how flexible and squishy these little hydrogels are. To overcome this, we developed a custom long fiber extruder for fresh printing. So we can extrude prefabricated fibers through the interaction of these two driving wheels into our extrusion needle, which then goes into that support bath. This allows us to do multi-material printing, where we can first print the hydrogel. In this case, we're using alginate, which is a hydrogel device derived from seaweed. And then we can switch print heads and embed these fibers, and then again print layers of alginate on top to fully encapsulate the fiber. The support bath can be liquefied in order to retrieve the part just by raising the temperature, and it essentially melts the gelatin microbeads that make up the support bath. Of course, this process is essentially trying to push a little bit of thread into Jello. And so the printability of any given fiber is going to depend on the fiber and the hydrogel properties. If the fiber is too compliant or the gel too stiff, the fiber will buckle. So we've developed analytical and computational methods for the printing process in order to allow us to provide a printability model. And we validated this experimentally across a number of fiber types and sizes. Additionally, we found that closed loop control based on computer vision actually allows us to kind of identify buckling early and adjust the extrusion rate to prevent print failures. One of the primary benefits from this technique is the ability to structurally reinforce hydrogels. So to test this, we conducted tensile testing of our prints with and without fiber reinforcing. And we found that, as might be expected, reinforcing significantly increased the ultimate tensile strength and tensile modulus of our composites. As one would expect, the reinforcement also improves the properties of these hydrogels in bending. So this technique, I think, is broadly applicable in soft biocompatible robotics. We've printed now with a number of different thread types, so little tiny metal wires, custom threads, polyesters, silks, single ply, double ply, triple ply, the collagen fibers themselves. And we can print them into both alginate hydrogels and collagen, and I'm sure many others as well. We can print one fiber at a time or multiple fibers, and we can print in 1D, 2.5D, or even 3D. And so we're now kind of working towards integrating this with the muscle tissue in order to create better transitions between the muscle and the robot and get better biohybrid robot outcomes. Okay. I'm going to change gears a little bit and talk about how we're looking at the neural networks in these systems as well. One challenge in working with muscle tissue in robots is that the muscle really performs best when stimulated via the natural neural circuitry. So if you just electrically stimulate muscle, it fatigues very quickly, and it has kind of low force capabilities. Whereas if you stimulate by motor patterns in the ganglia, it performs much better. So we're trying to understand how we can design neural networks composed solely of biological neurons to generate and program behaviors. To do this, we're starting by really quantifying the growth patterns of neurons in vitro. And so from this information, we can identify metrics that we can use to both drive neural models, but also validate neural models against experimental data. These include things like degree, total length, tortuosity, and we quantified this over a just tremendous number of neurons. The poor undergraduate students and Ashley spent a ton of time even just hand tracing these. And we can use these combined with some new morphometrics like change point test quantification, where we have a segment length and a number of change points, and find which metrics really tell us what phase of growth the neuron is in. These distributions can then be fed into excuse me, neural models to predict the formation of networks before we spend the time and resources needed to actually grow these in culture dishes. So now we're working on connecting these to those dynamical models, similar to those that you saw in the early part of my talk. Okay. To wrap us up, before I run out of time here, I'll just share a little bit about our recent work in biodegradable robots. So, it's very challenging to work with muscle tissue as an actuator, but the biologically derived materials have a lot of advantages for large-scale deployment in sensitive ecosystems, right? If we want to be able to release swarms of robots into a coral reef or into other fragile zones, we don't want them to get lost and break down and just be, microplastics and heavy metals. And so what we've been doing is adapting our alginate printing technique to create fully biodegradable robotic actuators. So we use that fresh printing technique that I introduced earlier, and we start by creating these CAD designs, which we can then print with the alginate in a gelatin support bath. The support bath contains calcium chloride, which cross-links the alginate, and we incubate the structures to allow that cross-linking to achieve kind of water tightness. The support bath is then liquefied, and we can recover the part. So we first validated this approach testing mini-scale bending and linear actuators. So note this scale bar is only 2 millimeters, so these are very tiny. And excitingly, these actuators show relatively robust performance over hundreds of cycles and can maintain their functionality even over tens of thousands of cycles in some of our testing. With this approach, we can build obviously the linear actuators, bending actuators. We can print wee little tiny grippers, and these grippers are able to pick up a wide variety of small, rough, fragile, and even slippery objects in aqueous environments. And we can also then print actuators that would be very challenging to cast, right? So here we've got kind of like a twisted bellows that gives you this almost elephant trunk-like motion. This can be used to pick things up or hook things. We can also print networks of these actuators. So here we have 3 actuators, each with their own pressurization channel to give us kind of positioning of the end effector here. And we've now used this to build wee little tiny robot arms. But of course, the I think most exciting feature of these types of actuators is that they're biocompatible, right? They can safely degrade in natural environments. And we tested this by introducing these actuators into our sea slug aquariums. And here you can see that these actuators degrade over about 7 days with our current cross-linking configurations. The actuators are even edible and digestible by marine invertebrates. So here we're feeding actuators to a sea slug and assessing that, yes, it has been digested. Because these actuators use calcium for ionic cross-linking, we can also do kind of rapid degradation. So if you needed to suddenly self-destruct your robot, you can introduce chelators, and the actuator will just come apart into alginate monomers. So, we're still working with a number of limitations with these actuators and trying to translate them so that we don't need an external source of pressure. But they're really helping push the boundaries of what we can achieve with biodegradable soft actuators. In fact, with these, we're seeing weightlifting capabilities higher than those currently seen in muscle-based actuators, while having similar degradation timescales. So we think this is a really exciting approach in biodegradable robotics. Okay, feel like I'm starting to run out of time, so I'll wrap up with some future work. We are really continuing to advance each of these fronts that I've introduced today. In the biodegradable material space, next steps are really focusing on packaging and life support for actuators to protect them from environmental contaminants, as well as scalable manufacturing. Right now, there's a lot of grad students with tweezers and pipettes building robots in my lab. We're also working on improving the force capabilities of bioactuators, as well as translating the principles from our muscle-based actuators into synthetic actuators with bio-inspired mechanics. We're continuing to work towards programmable living controllers made with biological neurons and adapting the dynamics and network structures from these biological neurons into biomimetic controllers. And we're starting to develop design tools when working with these strange materials. Robots that degrade is a very challenging design and control problem, right? How do you design a controller when the mechanics of your robot are going to vary over time? Can you leverage that even to do behaviors that you would otherwise not be able to achieve? And ultimately, our hope is that this all leads us to autonomous, biodegradable robots. So I'll just wrap up by acknowledging again the wonderful people who actually do this work, as well as our amazing collaborators and our funding sources. Most of this work that you've seen today has been funded by the NSF and the Army Research Office. And I would, of course, be happy to take any questions.
Thank you very much, Vickie. Very exciting talk. So let's open the stage for questions. You can either type in the chat or directly raise your hand and ask. Yeah, I mean, maybe I'll start with an icebreaker. I think it's very exciting to see that you're looking at design problems where instead of looking at particular tasks at one point in time, you're looking more at a trajectory of designs. And somehow you have to be robust to things that happen over time. And I think that's very interesting. What would you say is the best way to verify that these designs actually behave the way you want? So what's the way to go for you? Experiments, building simulators that really represent well what would happen? What do you think is the best way to estimate these long horizons you're caring about?
Yeah, I think you need both simulation and experimentation. We do a lot of modeling work. There are some fairly biologically plausible models of things like muscle and things like neurons. And so we can do a lot of prediction in silico, but we're always limited by that kind of sim to real gap, right? It's especially prevalent, of course, in soft robotics. And then we add another wrinkle to it when we work with the biologically derived materials because there's a lot of stochasticity. Muscle is not a motor. It's not going to respond exactly the same way every time you stimulate it. So we try to capture that stochasticity in the models as well, but you really need both. And it's a back and forth, right? You implement the model based on how you think the system works. You run the simulation, you get a design, you get behavior, and then you go to the experiments and you run the same experiment. And anywhere that the model and the experiment deviate, we like to talk about in my lab this idea of demand-driven complexity, right? So you try to validate the model. If it's not a great match, you look at why it's not a great match and you go add additional complexity there to address that. And you just have this iterative loop, right? Because we want our models to be kind of as computationally efficient as possible, as broadly applicable as possible, but still valid to our experimental platforms. So you need both. Yeah, it's a long way of saying that.
Okay, that's okay. I was imagining it, but I wanted to hear it from you. And in this context of iterative testing, so are you able to isolate effects of components at the system level, or essentially every time you switch a component, which could be a control routine, or it could be a physical piece of your robot, you have to essentially reevaluate the entire design? Because that's what overlaps with my research, and I would be interested about these kind of applications.
Yeah, sometimes there's a thing you change and you're just like, oh, got to completely retune the controller, right? But I think with this type of iterative approach, oftentimes, there's a lot of overhead to get that first kind of like nominal model set up. But once that exists and is at least qualitatively validated, as you're changing subsets of that model, oftentimes the changes to other areas are more minimal. But they still occur, right? So an example would be with the models we're doing for the sea slug feeding apparatus. we started with a really nominal model of the biomechanics, basically as basic as we could. And we've now got actually a series of more complex biomechanical models that'll be getting published soon. And as we've added those in, the students will have to go back and maybe add another motor neuron in the neural controller, or retune some of the synaptic weights because you've changed the mechanics. So it's a little bit back and forth for sure.
Okay, thank you very much, Vickie. I have another question, but first let's give the stage to other people in the audience. Subrata, you have your hand raised. Go ahead. You're muted, Subrata. Yes, now we can hear you.
Yeah, thank you very much. It was a great talk, and it's a very new subject and very interesting one. I'm interested to know what are the materials used by your team for the 3D printing?
Yeah, so for the there's a lot of different 3D printing used in my lab. Do you mean for the biodegradable printing at the end there?
Yes.
Yeah, so the support bath is made out of like a gelatin microparticles. So that gives us kind of a shear thinning effect, so that as we move the needle through it, it behaves like a liquid, but then it'll support what you've extruded into it. And then the inks that we're working with, so we work a lot with calcium alginate as our hydrogel. In order to print those structures, the support bath is washed in a calcium chloride solution, and then we extrude the alginate precursor, so the monomer mix, into the support bath. And then during printing, the calcium that's in the support bath starts cross-linking it. And after we melt the support bath, we do a post-processing step where we transfer the part into kind of a pure calcium chloride solution to finalize the cross-linking. There's other cross-linking chemistries that work with alginates as well if you need different mechanical properties. We also do use a very similar printing technique with the muscle tissues. And in that case, the support bath is still a gelatin microparticle based support bath. However, it's prepared under sterile conditions to protect the muscle from bacteria. And then our muscle inks, we work with both C2C12 cell lines and with primary muscle cells harvested from chicken eggs.
Great. Thank you. Thank you very much.
Thank you, Subrata, for the question. Maybe a final one I have is, so you're doing a lot of work on minimal robots for this set of applications. Do you find that some of these findings could be translated back to, let's say, more traditional robots that are mainstream these days, where minimality is oftentimes not the question there, right? But they would benefit from these kind of approaches. What do you think?
Yeah, so I think there's a lot of promise with the biomimetic approaches that we've been taking with our soft robotic structures. The synthetic nervous systems are very interesting, and I think there's applications there to be able to create and train sparser neural networks for robotic control that is not restricted to the types of robots that we've tested it on so far. There's also great work in the literature using synthetic nervous systems to control legged robots. So that could be a direct translation to more conventional robots. And then, I also see a lot of feedback loops from the biomimetic space where we're using these robots to understand biology into mainstream robotics, because as we learn more and more about what makes animal musculoskeletal systems so darn efficient, right, we can kind of pick and choose the features of that we then translate to robotic systems. So it's one of those things where it's helpful to have, I think, these kind of collaborative cycles with the biologists, the biomimetic roboticists, and the more conventional roboticists, so that you can really kind of be having these conversations in real time, so that it's easier to translate between those domains.
And that's great. I mean, I really look forward to seeing all these things join together, join efforts together. Oh, okay. I see there is another question from Subrata. Go ahead.
Yeah, very curious to know about the biodegradable robots. What is the normal lifespan of those robots in which you would like to see on the horizon of your experiment getting completed, just as an indicator?
Yeah. So there's multiple answers to this question. So just with the calcium chloride cross-linking, if we don't do any other cross-linking, those, if you just stick them in our aquarium that's filled with little microbes and snails and things that want to eat them, they stop functioning within a couple days, and they're completely degraded in 7 days.
Okay.
We have done some just like benchtop testing where we've used other cross-linking chemistries. So we'll go back in with like Genopin or EDC NHS, and those are more covalent bonds, so they're a little more robust, and those will hold up for longer. But we're still probably talking on the order of like weeks at most using these types of materials. One thing I'm curious about moving forward is getting into some more like structurally robust biologically derived materials, right? You could use keratins or chitosins or things like that. And that may extend them more while still being able to biodegrade. So I think it's a really rich space to try to work with other materials as well.
Great. Thank you.
Thank you, Subrata, for the question. And I take the occasion also to thank you again, Vickie, for the great talk. It seems that you have a great future directions as well. So I look forward to seeing the next results. So thank you again. I will reach out with the video once it's recorded. And thank you everyone for joining. I'll see you next week with the next Autonomy Talk.
Thank you so much for having me.